Hello all. So today we are going to see Morgan genetics, and we will also see that crossing over, linkage and recombination, how it will be extracted from Morgan genetics. Okay, so what is the organism he has taken? He has taken Drosophila melanogaster, which is fruit fly, common name, and the genes located in X chromosome. Okay, they already know that. So what is the experiment he did? Morgan experiment. We can see it. You see here, this is the one type of cross he has done. Okay, so male Drosophila he has taken and female Drosophila he has taken. So in body color you can see the brown body, which is given Y plus, and Y is your yellow body of the female, and eye color red eye dominant over the white eye. Okay, so now you can see here that then he has taken the female. So you see here, female is homozygous for both the genes, and it is recessive in nature. Okay, so male homozygous, but it is dominant for both. So Y plus is for brown body and W plus is for your red eye. Okay, so this is wild type. We call, in case of Morgan genetics, this is wild type. Okay, wild type. So now you see here how many types of gametes it will give. So we have to do the same thing that we have done in Mendel. Okay, we have to find the F1 generation, we have to find the F2 generation, and we have to see that whether we are getting the same ratio or not. What is the ratio of the F2 ratio? 9:3:3:1. Okay, so now you see here in F1 generation, so what we have to do, we have to take, you see here, how many types of gametes it will give. So in both the chromosomes, both are X chromosomes, you can see here YW and YW, so only one type of gamete it will give. Okay, so female will give only one type of gamete, okay, from the parental type. So now you see here, in this case, in case of male, you will get two types. One will be, this one will be your Y chromosome. Okay, this is your Y chromosome, this one, and this one is your X chromosome. Okay, so these two types of gametes you are getting here, and only one type you are getting here. Now in F1 generation, what you have to do, you are basically crossing these two. If you cross these two, how many types of gametes you will get here? You can see here that so you see here that here this one will cross with this. So you can see that YW and Y plus W plus. So this one will be female because both are X chromosomes. Okay, and this one will cross with this. Okay, so now you will get YW and this is Y chromosome, so male. So in F1 generation you got this one heterozygous for both the characters, you got the female, and male you got recessive homozygous for both the characters. Okay, so for now what we have to do, we have to self both. So for F2 generation we have to self all the female with all the male. So whatever the male, these are the types of male you got, not only one, you have to think like you will get some 300, 400 or 500. So you have to mate both. Okay, once you mate both, you have to understand one thing, that what are the types of gametes you will get from the female. So now see here, the types of gametes you will get here is the gamete formation. So meiosis will happen, so crossing over will happen. So I already put a video related to this, you can see that. So what will happen, so in this case, when the crossing over will happen, this part will go here, this part will go here, and it will separate from in the middle. So you will get YW plus and Y plus W. Okay, so how many types of gametes you are getting here? 1 YW, YW plus, Y plus W, and Y plus W plus. So 4 different types of gametes you are getting from the female. Okay, from the female. So from the male, how many types you will get? Male having, so Y chromosome is very small compared to actually X chromosome, so we don't consider that they are going for any crossing over, so we take as such. So YW will be 1, this is your X chromosome, and this one will be your Y chromosome. So now when you do a selfing between these two, okay, so what are the types you will get here in F2 generation? F2 generation, you can see here the parental female will be this YW will attach with this one first. Okay, so this is X chromosome from the female, this X chromosome is from the male. Okay, so what you will get, XX, one female you will get this type. Then YW plus from the female, and Y and W from the male. Okay, so similarly if you do for all 4, you will get this type, this 4 types of females. Okay, these 4 types. So we can divide it into 2 types. One is parental female, one is non-parental. Why non-parental? Because YW plus YW, Y plus W and YW, it is not there initially. These two are there initially. If you see here, you can see here YW and YW, Y plus and W plus. These two varieties are there initially in parental type. That is why it is called parental type. And these two are non-parental type. Okay, or recombinant type. Similarly, for males also you can do. So these are the 4 types. Okay, you got from gametes, you got from the female. Now for this X chromosome has to combine with the Y chromosome. XY will give you a male. So how many males you will get? These 4 types of male you will get. Okay, so these types are called as parental male types. These are divided into, this is your parental male type and these are your non-parental male type, YW plus Y plus W. So he did this and he got all these varieties. Now what is his expectation? Now you see here the findings are like, expectation will be like the Mendel he was expecting I will get 9:3:3:1 ratio. Okay, so as a dihybrid ratio, but he didn't get that. Instead of what he got, observation he got, the parental type is 98.7% and the non-parental type is 1.3. What is the meaning of that? Meaning is, means if you combine both male and female, this whole, this is your parental type. Okay, and in case of female, and this is your parental type in case of male. These are total 98.7%. Okay, and if you combine this is your non-parental type in case of female and this is your non-parental type in case of male. If you combine both, you got 1.3% only. So what he understood, he said that he gave a concept of linkage. Okay, what he said, the strength between these two genes are very high. So the strength between these two genes are very high because they are located very near to each other. That is why they are not undergoing crossing over very often. Okay, so then why we are getting 1.3? Because very little amount of crossing over is happening. So those varieties are called as recombination type. It is possible because of the crossing over, but low crossing over in this case if you are taking these two characters. So now what he finally said, if two genes are present at a distance to each other, it will be distance to each other will be distant to each other, then chances of crossing over will be very high. So recombinant varieties will be higher. Okay, if for example you see here one diagram I'll show you. So in this case actually what he said, you see this diagram. So this is the actually X chromosome. Okay, this is the X chromosome of the drosophila. So these are these many genes are there. Okay, so what he has taken yellow and white. Okay, yellow body and white eye or red eye. Okay, these are, so they are very near to each other. So whenever they are going under crossing over, they are moving together. Another experiment also he has done. In that case he has taken the miniature wings. So in that case the distance between them is very high. So he got very higher amount of recombinant varieties. Okay, I hope you are clear about now. So understand one thing, crossing over is required for recombination. If crossing over is happening very less, then recombination frequency will be very less and your parental variety will be more. Okay, thank you.